And welcome back, everyone. I hope you are refreshed. So you rejoin us for our fourth keynote session today, and then we will have what I hope will be a fascinating panel discussion on conducting impactful research. And then after that, the rest of the afternoon has in store exploring the Barbican and then some informal discussions. So we still have several items left to enjoy this afternoon. So I would like to introduce our next keynote speaker, Thomas Gorochowski. Welcome, Thomas. Thomas is Professor of Biomedical Engineering at the University of Bristol. We're looking forward to your talk. Thanks for being with us.
Thank you very much. Can everyone hear me okay? I can't tell. Yeah, sounds good. So firstly, I just want to say thank you to the organisers for inviting us here today. I think it's a really nice venue for this sort of thing. I find it really interesting wandering around, looking at some of the architecture and things like that. And as you'll see from my slides, the area I work in is a little bit different. So I've learned a lot today from all of you, so thank you for that. But to give you maybe a bit of context of where I come from and what my group is interested in, I originally trained as a computer scientist, and I worked in complex systems theory and was really interested in kind of emergent phenomenon that we see all over the place in technological and natural systems. But it was actually in a lecture that I actually got to see this video. And so this is a video of effectively an immune cell that's kind of hunting down this bacteria to basically engulf it and eat it and hopefully remove that from your blood system. Now, the thing that kind of fascinated me here was the fact that this cell is effectively, you can imagine it as just a pocket of chemicals whizzing around, interacting with each other, but somehow there was this kind of ability for it to make decisions, to move, to do all sorts of interesting things, which in my mind kind of sort of linked to kind of the ability to carry out computations. And so a lot of what my group looks at is how we can take ideas from computer science and actually embed them into biology with this idea of trying to reprogram biology across all the sorts of different scales that we see in nature. And so, like I said, it's this idea of thinking about things that biology can do, that we can then repurpose, but also augmenting it with new functionalities that it might not naturally do. And some of you might have heard of this field called synthetic biology or engineering biology, where we try and really bring to bear some of these kind of engineering design principles and ideas onto biological systems to actually kind of harness biology in new and interesting and hopefully useful ways. Now, the research in my group is really kind of centered at the moment around these three kind of areas or pillars, which sort of help us tackle that at different scales and looking at different aspects of that. So there's a specific area that looks at trying to design what we call synthetic genetic circuits. So these are effectively pieces of DNA that we can put into cells and will alter the gene expression that's going on within them and ultimately the phenotype or the function that that cell is performing. So we need kind of components we can stick together in different ways to do that. Another is kind of looking away from this kind of cell-centric view to kind of a community view. A lot of behaviors in biology are at the collective level. And so trying to understand how collectives of cells make decisions or assemble to form certain structures is something else that we're very interested in. I'll show you some examples of that. And the final part, which I'm not going to talk about today, is something that over, I'd say, the last 2 or 3 years we've become really interested in. And it's this idea of trying to engineer aspects which are really unique to biology in my mind. And that's this ability for adaptation and evolution and ways that we can actually guide that process in living systems. So not necessarily carrying out evolution on a computer and then, I guess, fabricating that in biology, but actually fabricating a biological system that has certain variational properties that we want to kind of guide the evolutionary processes that are present. But anyway, to start, I want to begin by giving you a bit of a kind of a tutorial on how we actually go about reprogramming individual cells and some of these ideas around genetic circuits. So this is kind of one of the most beautiful pictures, I think, in biology. This is a kind of an illustration by David Goodsell, who's now retired, but he was previously at the Scripps Institute in the US. And I think it shows you the kind of the complexity of a cell and actually how packed and crazily complex a cell actually is. Now, the sorts of circuits that we build are basically, like I said, these DNA molecules that we design on a computer, but then when we get synthesized, and they go into this kind of pool of messiness that is biology. Now, in order for those circuits to be executed, they go through a number of kind of very basic processes, which is known as the central dogma of biology. So this DNA, first of all, it's read by these machines called RNA polymerases, which transcribe that DNA into RNA. And then that RNA gets translated into proteins by ribosomes. And it's those proteins that typically make up the kind of core machinery of the cell. Now, as a computer scientist, when I first saw these sorts of images and sort of biologists were talking about all this stuff that was going on in cells, I was kind of horrified. Like, how on earth are we going to potentially get the cells to do anything? There's just so much noise and stochasticity and just diversity within these sorts of systems. And so what we've had to do is to basically abstract away a lot of those details preliminary, just to be able to make a kind of a tangible start on actually controlling the behavior of these sorts of systems. And so what we typically do is we ignore many of those levels and we just focus precisely on how the DNA itself is actually being read. So to do that, what we can do is we can actually make use of sequences that we know we find in nature, which actually carry out very specific functions around how DNA is read by a cell. So for example, there are these sequences called promoters, which basically tell RNA polymerases where to bind and start transcribing the DNA. And there are similar sequences called terminators, which basically signal to those RNA polymerases to stop transcription. And so what we can do is we can actually design sequences that contain these kind of known parts to actually guide where there are flows of transcription and how that DNA is actually being read. Now, the interesting part is that we can also encode within this DNA other sorts of molecular machines that can then go on and have other kind of effects on that particular circuit. So in this case, this sort of symbol here is denoting what we call a coding sequence. This is encoding a sequence for a protein, which is then able to physically bind this promoter sequence here. And that obviously then stops these RNA polymerases being able to bind to that particular part of DNA, and it turns off transcription of this region here. And so what this allows us to do is to basically control how flows in maybe one part of the DNA are actually affecting flows in another, and actually implement some very basic kind of logical operations that we would use in, say, computer science for carrying out certain types of logic. So in this case, this would be acting a little bit like a NOT gate. Now, one of the nice things about this kind of abstraction is that we have a kind of a common signal that we're using for propagating information through these systems. And that's this flow of RNA polymerases, which is ultimately controlled by these promoters. So we can actually build more complex circuits just by making the output promoters of, say, one of these gates the input promoter to the next. And that's been done a lot in the field to create higher order circuits that then carry out maybe more intricate or more interesting functions that we might want. So this is just an example of what's known as a repressolator. It's basically 3 genes that inhibit each other in a cycle. And if you have an odd number of genes, then what you end up producing are effectively sustained oscillations in gene expression. And so you could imagine this acting a little bit like a clock in the cell that you could then sort of tap into to do various things. Now, this all sounds really good, and I've shown you these lovely illustrations and animations of how this stuff works. But the problem that we've faced in the field is that while we can create these sorts of systems that maybe contain, I don't know, a handful, maybe tens of different genetic components, as we start to scale up the complexity of these systems, what we typically find is that there are lots of aspects of the biology that we just don't understand well enough, and the systems just fail to work, so they break. And so this is an example of some of the ways in which they break. So we sometimes find that actually the sequences that we use have multiple functions that we're unaware of, and so that can cause issues in terms of kind of transcription in regions of the DNA that we don't want. Sometimes parts just don't work. Our knowledge is limited, and so we just don't know why, but they just don't seem to function in certain contexts. Another, which is really prevalent in genetic systems, is the fact that DNA sequences and the function that they encode is often inherently linked to sequences nearby to those sequences. And so you can have the same part, and by that I just mean DNA sequence, which has slightly different sequences nearby, and that can cause a big change in the actual function that that part then displays in the system itself. And then finally, something that I've completely ignored in those other examples is we're putting this into a living cell that hasn't evolved to actually run the function that we're interested in. And so there are often these interactions with the host cell, which often cause the system to break. The cell tries to turn off these kind of burdensome, energy-sapping processes that we're kind of installing into them. Now, these sorts of failure modes are also present in other types of engineered system, but unlike in, say, electrical engineering or computer engineering, where we have tools like multimeters or debuggers or profilers to actually interrogate what's happening within our programs, in biology we often don't have those tools available to us, and so we're unable to actually observe what's happening at the level of DNA to know why a system might not actually be functioning. And so one of the things that my group has been doing over the last nearly decade now has been trying to adapt sequencing methodologies, some of which we already saw in some of the previous talks, to try and open up a window onto how these kind of larger genetic systems are actually functioning by basically reading off some of the components that get produced. So you can see these little grey lines that are emanating from the DNA. That's meant to represent the RNAs that are being transcribed, and we can use a technique called RNA sequencing to read those and pile up those reads to generate profiles about the density of RNA polymerases across that particular piece of DNA. Now, to show you what that looks like in reality, this is a kind of a fairly simple decision-making circuit. It's just a logic circuit which has 3 inputs and a single output. And at the time when we built this was probably one of the largest synthetic genetic circuits that have been built that kind of worked. What we can do is we can take a kind of a logical description like this circuit diagram. We can then compile that down to a DNA sequence by using the motifs, or some of the motifs that I showed you earlier, and the fact of how we can connect these various parts together to produce a DNA sequence like this. And then we can put that into a cell and see what happens. And so what these distributions here are showing are basically for each of the 8 different combinations of those inputs. In grey, we're looking at effectively the fluorescence that cell is producing. And the reason we're looking at fluorescence is that this final protein here is a yellow fluorescent protein. So if the cell is switched on, it'll glow yellow, and if it's switched off, it won't glow at all. And what I've overlaid on this actually as well is actually the model that we use for building these circuits underlying it. So what you'll kind of see is that for some of these states, actually our model and the experiment tie up really, really well. But what you start to see in some of these latter states is that there's a larger deviation. Now, if we look at just these distributions here, there's no way that we can know why these deviations are happening, because there's over 40 components here that could be faulty, and there's just not enough information contained within these distributions. And so what we can do instead is that we can use RNA sequencing, as I said, to produce these what we call transcriptional profiles to give us some insight into how transcription across that entire circuit is varying across some of these different states that it's being exposed to. And so that's exactly what these are showing here. Again, I've overlaid in red and blue the kind of the model predictions. And we can then take these distributions, these profiles, and effectively use simple models to interrogate how increases and decreases correspond to some of the basic parts I mentioned previously, so these promoters and terminators. And by looking at how the kind of the strength of those promoters is varying across these different input states, we can then actually generate effectively characterizations for each of those components within the context of the circuit itself. So for the sensors which act as the inputs to this particular circuit, we can look at their off and on states. And so here, these dashed lines are effectively what the model that we've used to build this system is saying these levels should be. And these kind of solid bars are showing kind of what we actually measure from the system itself. And then similarly, for some of the logic gates that we're using, we can look at the transfer functions or the response functions to see how the inputs and the outputs are varying experimentally and how we would expect them to vary in the model itself. Now, the beauty of this is that this gives us the information that we need to try and pinpoint where the failure is actually occurring. And in this case, you might have noticed there's quite a big deviation here between this kind of on state and where it actually should be. And what we can then do is we can go in, we can replace, it's actually this promoter here that's faulty, replace that with a different sequence which has a similar function, but maybe a different sequence which isn't going to have any contextual effects, and actually recover the function that we want for that particular system. So what I'm trying to show you here is that we have a lot of data in biology, like there's no question about that, but often we don't have the right data to make the right decisions. And so some of these sequencing techniques can actually give us the right sorts of insights to actually make the right decisions when we're actually designing some of these genetic systems. Now, as I said earlier, obviously I've ignored there all those other processes that I described of what goes on in the cell when a piece of DNA is kind of introduced. And so one of the things that my group has been doing kind of more recently has been trying to adapt other sequencing methodologies to effectively give us information about some of those other processes that are taking place. So there are methods like ribosome profiling, which I won't go into, which allow us to understand how ribosomes are actually translating those RNAs into proteins. We've also been using techniques called like nanopore sequencing, which allow us to also look at modifications that can be made to some of these molecules. And those modifications can be really important for regulation, which is key to these sorts of decision-making circuits that we're building. So we're really collecting here a wealth of data, all collected from kind of sequencing technologies, but that are giving us very different insights into what's going on in these cells. And I guess I'm putting this up to kind of hopefully advocate some interest in some of you. You might see some ways that you could use some of the kind of the multimodal AI techniques that you're developing to bring together some of this information to better understand how these systems are working, or potentially to generate new systems with interesting functionalities based on that. Now, I realize I'm at a multimodal AI conference. I haven't said anything about AI. I've just told you about some experiments that we do and some of the things that we build in the lab. The reason that, I guess, as a bioengineer, I'm very interested in kind of AI and in particular machine learning is the fact that as a bioengineer, although we can write DNA now quite effectively, that's not the exciting bit. The exciting bit is the phenotype, the actual function that the cell carries out, and those are the things that we want to design. But the issue is that we don't always know what genotype, so DNA sequence, to write in order to achieve the phenotype that we're interested in. And these experiments are really slow. Like building those circuits that I showed you previously, when we built those, that was around 2014, it takes months to get the things constructed and verified and working. And so the thought of running thousands of different designs is just ludicrous. It's not gonna happen. And so the hope's always been that there might be ways of using different machine learning techniques to kind of understand this relationship better, such that we can actually, rather than having to always go slowly through experiment after experiment, we can perhaps even provide a phenotype that we want, and it can then generate a sequence which it thinks is actually going to give us that. And I think we've already seen some examples of that in other fields today. But designing DNA sequences is very hard because it's an absolutely massive space, a design space that we have to kind of work with. Now, a couple of the areas that we've been looking at specific to this problem is around how do you potentially collect suitable data for training these sorts of machine learning models? Because that's one of the things my lab does, is we generate these really interesting, rich data sets, but are they the right ones? Like, how accurate can these predictors become? And secondly, like, what kind of models should we be using in these systems to actually make them useful? Now, I think I've already probably shown you this, or explained this in a different way, but one of the things that gets me very excited about sequencing in particular is just the range of different things it can tell us about biology. So I've kind of talked a lot about kind of aspects of the central dogma that we can measure, but there are other sequencing techniques that can give us information about molecular interactions. We can understand how proteins and DNA are interacting, which is really important for building these kind of regulatory circuits. There are other methodologies that can even tell us things about the structure that some of these molecules are making. So in terms of shape-seq, looking at RNA structures, which are also really important to their function. And it can even do that with dynamics. So you can understand how these structures are forming over time, which I think is amazing. And lastly, there are these very high throughput genotype to phenotype kind of massively parallel reporter assays, which I think the last talk really touched upon really nicely, around understanding how particular genotypes map to particular phenotypes, but producing potentially millions or tens of millions or billions of relationships that you can then use to train these kind of machine learning type models. And so fundamentally, I think sequencing, it's not the only, but it's a really powerful way of generating data sets which can cover a whole diversity of kind of biological modalities that we will need to bring together in order to actually engineer biology effectively. Now, one of the kind of, I'd say one of the most valuable methodologies here for kind of machine learning, as I said before, are these kind of what they call massively parallel reporter assays, or MPRAs. And these are typically based around kind of quite a complex experimental process. So I'll kind of walk you through this. So there's an initial step where you have to actually synthesize a library of different sequences. So this would be your designs, your DNA designs. And this could be on the order of thousands or tens of thousands or maybe even hundreds of thousands of different sequences. And the important thing here is you have to couple that sequence to some function that you can then measure en masse. And in typical MPRAs or flow-seq experiments, which I've shown here, is typically coupled to the fluorescence of a fluorescent protein. So here I've used green fluorescent protein as the kind of the example. So what we're effectively able to measure, if you took a single cell with a particular sequence within it, we'd be able to look at how green that cell is to understand the function that that particular sequence has. Now, in order to measure enough of those actual sequences, what we use is a machine called a cell sorter, which is able to then, at very high speed, effectively sort millions of cells into discrete bins of certain levels of fluorescence. So there's a discretization here in this, maybe a continuous space of fluorescence. We're discretizing the fluorescence of each cell into one of these bins. And what we can do then is we can take the cells from each of these bins, we can amplify the DNA, and we can sequence it to find out, well, what was the genotype that actually led to that level of fluorescence. And that then allows us to effectively, for each of those genotypes, generate discrete distributions of the fluorescence or the function of that particular sequence that we're interested in. And then the final part is we need some kind of inference step where we can take that discrete distribution and maybe fit some kind of distribution over it, continuous distribution that we can then use to kind of characterize what the strength of that particular component is. Now, the issue experimentally is that there are a huge number of parameters that you can choose when you do these experiments. And if I'm honest, a lot of the experimental data sets that exist, I've just tried to, this hasn't been chosen necessarily in a very systematic way. And so there's a lot of concern around whether the choices of parameters matter, whether they've picked the right ones, and even how accurate are these inferred values going to be based on the fact that there's a lot of steps that these kind of experiments have gone through. And so one of the things that we've been interested in is, well, how do you design these sorts of experiments to ensure that you can actually infer accurate values of the data that you're getting out the other end? Because that's going to be key if you want to use this for, say, some kind of machine learning based model. And so one of my students, Pierre, a while ago now, he basically set up a in silico simulator that tried to capture the kind of stochastic effects that can happen at each of those different stages. And then we could start with a known ground truth of distributions of functions that are kind of biologically realistic. We can run them through that simulator. We can carry out different forms of inference to see if that makes an effect, to then see how accurate the predictions are from that. And the nice thing about this is that we can also then actually use real flow-seq data with these inference methods to actually try to characterize more formally some of the data that we have available. So by using this kind of in silico platform, he was able to kind of show that basically how the error in the measurements that you're getting out at the end are related to different parameters that you have when you actually design these experiments. And I think the really beautiful thing about this was that it allowed us to come up with some very simple kind of rules of thumb that if you adhere to, mean that the data that you're going to get out is probably as good as you can expect. And so in this case, if you've got a library with, say, n different unique sequences, then you need at least 10 of these bins, typically logarithmically spaced, that each contain at least 100 cells and 100 sequencing reads per design in order to get an accurate inference of what the performance of that particular design actually is. So this is important because it's no good, like I said before, it's no good having lots of data if it's not of the right quality or the right form or looking at the right thing. And I think these are the sorts of rules of thumb that need to be kind of more propagated throughout the field itself. So we've used this type of approach to actually curate a number of kind of what they call large MPRA data sets that typically focus around kind of parts that are used to control translation of genes. And what we've been able to show is that by using these kind of refined data sets, where we're using kind of proper inference methods and we're kind of understanding the error that we can expect in the experiments based on the parameters under which they were carried out, that we can create typically hybrid convolutional recurrent neural networks that are able to predict kind of the strength of sequences with state-of-the-art accuracy. So these are as good or better than the kind of the mechanistic models that people typically use, or other deep learning type models that have been historically used. The issue that we face though as a bioengineer is that these sorts of experiments look nothing like the context in which we actually want to use these parts. And what we find when we actually take these models and then use them to predict how a part will function within a circuit that we're wanting to build, they typically break. There are other contextual aspects which mean that the predictive actually just goes down. It doesn't work as we would want. And the reason for that is that biology is really context dependent. So we can have a single sequence, but depending on, like I said, the nearby sequences that are present, the particular cell that sequence is in, the environment the cell's grown in, the sorts of food that you're providing to the cells, or even the actual experimental equipment you're using to measure it, they can all affect the measurements or the performance of that particular part. And that makes it very difficult in order to have a single model that actually captures the performance of that sequence. It doesn't really exist. It doesn't mean anything in a lot of ways. And so one of the things that we've been trying to do is to adapt transfer learning or fine-tuning type methodologies to allow us to train one of these deep neural networks with one of these very large data sets, which cost a lot of time and money to produce, and then effectively take the learning that model has and transfer it into a new domain, which is in the context that we actually want to use this particular DNA sequence in. And so if we take a few measurements of different designs in that context across the set of designs that we have, we can then refine the weights that we have in these models and then regain the predictive accuracy for the specific use case that we have. And we found that this can work really, really well. So for example, this is just an example of one of the places that we've used it, where we've taken a kind of effectively one of these deep neural network models that was trained on one of these data sets, but then we've used it in a completely different genetic context with a different strain, different conditions, and a different experimental setup, which obviously doesn't have great performance in kind of its original state. But if we take just 90 new measurements from the new context, we can then fine-tune and sort of transfer that learning into that new context that we're actually interested in. And that 90 numbers is nice for biologists because they have these plates that have 92 wells, and that's like a format that they can get on board with. And it's a number of experiments, which isn't too many to do in order to actually make a model that's potentially predictive. And that's obviously orders of magnitude less than if you had to train a model from scratch in that new particular context. Now, one of the other nice things that we found when we were doing this work is that those 90 measurements also can be chosen cleverly. So we could just randomly pick sequences and test them in the new context and see how well they do. But what we actually find is that if we actually use the model to pick examples that span the kind of the levels of performance that we might be interested in, we can actually improve the error rate in the prediction quite significantly, so that we again need less experimental measurements to actually produce a model that's tailored for the specific use case that we're interested in. And I think this is going to become really important because there are so many contexts in biology. The idea of a foundational model for some of these systems is going to be very difficult to achieve with the data sets that we have. And so by being able to fine-tune or transfer aspects of what's been learned is going to be really, really important, I think, moving forward. Now, moving away from these kind of small genetic systems, another place that we've been using kind of machine learning or AI type approaches is in looking at genome design. And in particular, the area of genome design we're interested in is minimal genome design. Now, the reason for that is that minimal genomes potentially will give us kind of some insight into kind of the minimal components needed for life. But from a bioengineering perspective, it also strips out a lot of unnecessary processes in cells which are needed in, say, a natural varying environment, but wouldn't necessarily be needed, say, in a fermenter where we're controlling the environment very carefully. And so the idea is that that could free up additional resources or abilities for the cells to actually perform more or better under certain conditions. Now, one of the difficulties with thinking about doing genome design is that you have this thousands of genes that make up a genome, and like how do you know which ones to potentially remove? Especially because to do that experimentally, it's just infeasible. It would take too long. And so to get around that, one of the approaches that we've used is to make use of what are known as whole cell models. So these are very, very large mathematical models. They're stochastic. They have tens of thousands of equations, tens of thousands of states as well. And they try to capture all of our current understanding of what goes on in a cell. So this covers things like gene expression, metabolism, growth, some of the biophysical processes like genome replication. All those sorts of things are tried to be captured. Now, obviously, doing gene knockouts is going to affect every aspect of the cell, and these models are therefore able to capture some of those kind of nuanced effects that might occur across the sort of different processes that take place in a cell. And so they're really a great platform for kind of exploring some of those ideas. The main problem with them, though, is that they are very, very computationally intensive to run. And so it's still very challenging to use them if you want to test a large number of different genome designs, is the fact that it's going to take a long time in order to run that. Now, when we're thinking about minimal genomes, one of the other things that we need to think about is, well, what is a minimal genome? What does it mean? And in this context, what we've really thought about is basically the smallest number of genes that allows for a cell to remain viable. So that means it's able to replicate. But because some of these processes are stochastic, we can't just look at the simulation of, say, a single cell. We need to think about the simulation of cell over multiple generations, because there can be effects that are only seen later on in terms of its life cycle. And so what we're interested in is basically whether or not a cell is able to replicate, in this case, over 6 generations, as we end up with 32 different cells. There's 32 simulations that we have to carry out, or whether there are certain replication steps which don't actually work correctly. Now, if we were to actually carry out this sort of simulation using these whole cell models, it again would become completely infeasible. And so we needed a way of speeding up this ability to go from, say, a particular set of gene knockouts to a yes or a no, does the cell remain viable? And so the approach that we took was to try and see if we could make use of I think the term emulator was used earlier, or surrogate. We used that sort of terminology, a surrogate to capture the kind of the critical information or some of the relationships in some aspects of the states of a single simulation as to whether it would then remain viable over those full 6 generations. And so what we ended up being able to do is we took simulations of just the first, effectively doubling time of the organism. We then took a subset of those molecular states within that cell, and then we effectively took whether or not that cell was able to carry out then a further 6 divisions or not, generated a training data set from that, such that we could then, if we had a new set of gene knockouts, we could then just carry out just a single simulation of the cell to replication, use this surrogate to then decide whether or not over those 6 generations it would actually remain viable. And by taking that sort of approach, what we're able to show is that we can generate a fairly accurate surrogate model. In this case, we used just a random forest type approach that's able to actually quite accurately predict kind of small numbers of knockouts beyond the kind of the training or the training set that was used. But the issue we still faced was that as you start to have smaller and smaller genomes, the accuracy of this surrogate decreases quite significantly. And so what we ended up having to do was to basically retrain this surrogate at different steps during our minimization process in order to ensure that we're able to keep accuracy at a sufficient level. And so in the end, we incorporated this kind of approach into an approach which is known as Minesweeper. It's effectively a divide and conquer type gene design approach. And what it does is it effectively starts by classifying genes as either essential or non-essential. And we could use the surrogate in order to do that. And then we could effectively start removing small segments of genes of differing lengths and actually use the whole cell model and the surrogate to then make predictions on whether those cells remain viable. And then what the algorithm does, it starts combining some of those kind of larger chunks together to try and find effectively a smaller genome, or the smallest genome it effectively can. Now, when you go through this process, what you end up with is effectively a genome that has 737 genes that are modeled within the model itself. And that effectively is about a 40% reduction in the original model. What's really interesting is that, so here this MS56 is one of the experimentally created minimized genomes. It's not a minimal genome in any sense, but it's been minimized. And what's interesting is that actually there are effectively 383 genes that our genome design is saying could be knocked out in this organism, which currently aren't. So there's potentially some really interesting targets for us to look into. But the key thing here is that this became feasible. This approach of using this surrogate and retraining it allowed us to speed up that kind of genome design process 14-fold. So I think it took about, I think it was 6% of the original running time in terms of the actual computational power that was needed. And that's really important when you're thinking about these kind of large-scale design problems that we have in biology. So far I've spoken a lot about kind of genetics and kind of individual cells and this sort of cell-centric view. To end, I just wanted to sort of also talk about some work we've been doing around kind of thinking about controlling living collectives. And as I said before, this is because often in biology, a lot of functions emerge beyond the level of a single cell. So things like bet hedging or swarming, learning is a great example. We're in an AI conference. Like that doesn't happen necessarily just at the level of a single neuron. You need lots of them interacting in order for that to happen. But the problem we have in biology is that it's very difficult to study these kind of multi-agent systems. And so one of the things that we've been building over the last kind of 5 or 6 years is a system called the DOME. This is a very low cost, hacked together system which allows us to use effectively a microscope to image a kind of a small micro population that we have on our system that we place here in the sample, so it's just down here. But it also integrates a projector, so we can use light as a kind of a way of interacting with that population to shape the behaviors in interesting ways. And projections and light is really useful because we can pattern that in space and time, which gives us a lot of abilities to interact in interesting ways. Now, to kind of demonstrate kind of the sorts of things we've been doing with this, we've been taking what we'd call pond life. So if you probably, I saw there was a very green-looking kind of pond outside. I suspect some of these are living in there, no doubt. But we took some of these. These are the kind of motile microorganisms that typically are responsive to light. And we can place them on our system, and then we can start to expose them to light patterns to see what they do. So in this case, what you'll see is that some of the organisms, they get halted, but then what you often find is there's an adaptive response in some of these organisms, so they'll start moving again. Others are completely repelled by the light. Others do other things. But the reason for that this platform is quite interesting is that we can build it into a kind of a larger kind of sort of a screening, I guess, workflow, where we can take microorganisms, we can put them onto the dome, and then we can, because we have this projection system, we can start exposing them to time-varying light signals, spatially varying light signals, and we can use the data from the changes in their behavior to effectively fit generalized models of their behavior. And that isn't just at the level of the entire population, it can be at the level of the individual cells themselves. And so we can then use that to effectively create what we call a digital twin of that collective, which can act as a really powerful, I think the terminology is gym, that you could in theory train other sorts of algorithms on to understand how you might control or shape various aspects of the population or the collective's behavior. And to give you a very simple example, we've used those models to develop, in this case, this is an open loop type control methodology, but hopefully you can just about see BCL there, just about? So this is the biocompute lab, this is our initials. This is really crude, but what gets me excited, and one of the reasons I think I would love to speak to more of you, is that this sort of setup is kind of really prime for some kind of active learning, reinforcement learning type approach, where you could start to kind of refine the spatial behaviors that these organisms are actually producing. And I think there's a lot of scope to kind of to sort of look into those sorts of areas of research. So to end, in terms of take home, hopefully I've kind of demonstrated to you that actually engineering biology, AI is one of these kind of revolutionary technologies. Engineering biology is also a revolutionary technology. And I think bringing them together is really, really exciting, but it's really hard. I think that's the one thing hopefully I've also shown you. It's not easy to do these things. I think we have a lot of high throughput data-rich assays, and so we can measure a lot of these different aspects of what these systems are doing, but making sure we're measuring the right things is also going to be really, really important. And as I said at the end, although we have what I would call large data sets, they're probably not large enough for some of the sorts of AI approaches that some of you might want to use. And so I think we need to have these kind of active, kind of lab-in-the-loop style systems in order to produce an effective way of actually engineering some of these biological systems. So modal AI will be key. So I think you'll all be in a job, which is great. I also just wanted to highlight what I've shown you here is really the tip of the iceberg of the sorts of things we work on. There's a lot of other areas that we're very, very interested in. So if there's anything here that you think, oh, I'd love to know more about that, or I'd like to work in or collaborate on that, please let me know. And to end, I just want to thank, I don't do as much of this work as I used to, and I just want to thank all of the students and the collaborators and funders for making it possible. So I'd be happy to take any questions.
Firstly, how do we construct a target between regulation and then overregulation? Because overregulation can also be a problem. And then when you were talking about, for example, the context of for cells and biology, I know a lot of times when sometimes if you make a change, it activates one pathway, but then another pathway is also activated, which creates synergistic combinations when they're used together. So how do you anticipate us predicting that, especially when there's not a lot of information on what exactly would create a synergistic combination, and then how would we incorporate that in the greater context of trying to create a model that kind of looks at this?
Yeah, and I think it's a really good question. I think you kind of tapped into something that I think is quite important. The challenge we have in biology is that there's so many bits of it that we just do not know about, and so it's kind of it's an impossible task. How are you going to make a model of that when you don't even know what the things that it can actually potentially do? But I think that's one of the areas where engineering biology is actually going to open up some new avenues. So rather than say historically where we might take a natural genome, we might try and study it to understand what it does, but there might be a lot of contingency, which are sorts of things that you're saying about which we just don't understand the history enough to know why it's the way it is. But with engineered biology, we could develop these minimal genomes. In theory, would just be enough for the cell to work in a particular way, and so they're much less likely to have some of these kind of strange behaviors, which may be important for the biology in its natural native habitat, but might be really detrimental for trying to make it an engineering-like substrate that we can do things with. So I think where the power of this engineering biology comes is it allows us to perturb or create often simplified systems or insulated systems that get us away from some of those problems that you're kind of, I think highlighting. You mentioned about kind of overregulation and things like that. A lot of the the kind of models that we use for these sorts of circuits at the moment are very naive, and they often don't take into consideration the host and other things that are going on in the host. But there is a real movement to try and incorporate those sorts of aspects into the design process of the systems themselves. So one of the key things that you often see in these models is that they use up energy from the cell, and they use up resources. And for bacteria, machinery like ribosomes, which make proteins, are like really critical, because if a bacteria can make more of itself more quickly, then it's going to be selected for in the population. And so if you start taking away its ability to make its own proteins, it gets very unhappy, and you get selective pressures against that. So having models that understand how ribosomes are being distributed between the native cellular processes and the synthetic system are helping us understand, like, where is that point where we're not getting to the point where we're taking too much away that it just breaks, or there's some selective pressure that we don't want. So I think a lot of it, yeah, hopefully that answers both of the questions. I think there's the engineering aspect of this will open up simpler models which maybe mitigate that. And I think, yeah, just having more comprehensive models of cells and the important processes will get around some of the other issues.
Hi, thanks for a great talk. So my question was to do with I think a few slides ago you had the framework with the microscope and then the light. Yeah, that's the one. So, when you're shining the light, you're seeing if it's photokinesis or photophobic, and you have quite a few of these sort of microorganisms in the sample, right? How do you know when the microorganisms are in sort of a stable or an initial state such that you can be sure they might be photophobic? Or because I think on the iceberg slide you had sort of adaptive materials, right? So then that might be quite difficult seeing an initial state, for example.
Yeah. So yeah, again, it's quite hard. What is a base state of a cell? Is because if it lives in the sun, it's probably not in a dark microscope room or something like that. So it's a very difficult question. The thing we can do with this, at least, is control what the cells are going through. And actually, one of the reasons that, so here I kind of mentioned these sort of time varying inputs. A lot of the adaptive responses we see have a kind of a time scale, and actually by varying kind of the length of the pulses, we can actually pinpoint very specifically how long that adaptive response is. And once you know those time scales, it then helps you do all these other things as well. So there are aspects of just having the control over the environment which helps you at least understand, not necessarily what the baseline level is, but it helps you understand some of the aspects of these responses that are actually critical for having a model that's going to be predictive under a wide range of different conditions that you might expose the cells to. Yeah, the other point maybe to mention with this, I don't know if it came across, but we actually generate these kind of models for each individual within the population. So they're separately parameterized. So we're not getting, say, a single parameter for a population. We're getting distributions of parameters that are also co-varying in specific ways. And that's really critical for it to actually be representative of what you see a population doing, because there is a lot of heterogeneity across the cells themselves.
Cool, thank you.
Thank you. One last very quick question, please, and then we'll move to the panel discussion.
Thanks for a lovely talk. Another question on the light-controlled microorganisms, if that's all right.
Yeah.
I was just wondering, when you're trying to model the way that you're controlling them, are you treating it as a collection of agents, or are you treating it as kind of a probabilistic distribution of density?
Yeah, so we've got some work we do both, basically. So for certain control, so the control algorithm that we use here, we're just considering a density. We're not looking at each individual agent. We're looking at kind of, we want certain density, high density here, low density here, and that kind of thing. But there are some really interesting control algorithms that look at the individual cell level. And we've got in silico results that show that you can heavily refine the accuracy of the edges of these systems if you start going down to single cell control. So it seems like the using kind of a continuous approximation for density across these things can get you to a certain point, but in order to refine that, you then need to actually control at the individual level in order to get various things. So I'm saying we do both, but here it's just we're thinking about it as a density. But it's a really interesting control problem. And it is, my view is that this could be, there must be some kind of reinforcement learning type approaches you could apply to this as well, to learn what might be the right sort of control actions to apply, because it might be that those vary over time. Like it's not the case that you come up with a control law and you just run it and it works. It might be that that needs to vary because the populations will vary, the temperature in the lab varies, and they behave slightly differently. So being able to have that kind of system that can adapt will be really valuable for some of these applications.
Awesome, thanks.
Thank you very much indeed for a fascinating talk. Great to see your transition